Hi, good morning, and welcome to Grand Rounds. This is the last Grand Rounds lecture of the current academic year. In a couple weeks, we'll have my semi-annual town hall. But otherwise, as you'll all know, we try to invite a particularly distinguished speaker both to end our Grand Rounds for the year and also to kick off the Grand Rounds of the subsequent academic year. And this year, that will be the first or second Thursday in September. And with that, we're really so delighted to have Helen Mayberg with us here today. And I enjoyed catching up with Professor Mayberg briefly before her lecture this morning, and also really enjoyed reviewing her remarkable CV. In catching up with her just now, I learned that her favorite teacher when she was a trainee was our own Carl Braun. And we're delighted to have Dr. Braun with us today to introduce Dr. Mayberg.
So I want to welcome Helen Mayberg to Mount Sinai West and Mount Sinai St. Luke's, and specifically back to New York City, where she began her neurological career as a resident at Neurological Institute of New York, and that's where I first met her in 1982. Her energy and enthusiasm were infectious, and with Helen on neurology rounds, things were seldom dull. She had the capability of thinking out of the box. The work that she's accomplished in the localization and mechanisms of depression, first at Johns Hopkins, and then at the University of Texas, and then the University of Toronto, and for the last 13 years at Emory, placed Helen firmly in a long line of neurological investigators of brain function that started with the localizers, Broca, Wernicke, and Hewlings-Jackson in the late 19th century, to the neurocircuitry investigators, Sherrington and Eccles, in the first half of the 20th century, and to Norm Geschwin and others in the late 20th century, who brought neurology and neurological investigation back to localization of brain function. And these neurologists were largely involved with issues of language and memory. But there was another neurologist of the 19th century from Austria who traveled to Paris to study with Charcot. His name was Sigmund Freud, and as a newly minted neurologist, he was at the time convinced that specific emotional disorders could be localized to certain specific parts of the brain. Well, as we all know, he subsequently left Paris and left Charcot and returned to Vienna and Breuer and the greener pastures of Anna O and the subconscious. The resulting split between neurology and psychiatry has lasted almost a century. With Helen's work, I would suggest that we are back at the translational frontier of the application of brain localization and neurocircuitry to the spectrum of depressive disorders. But as I think you'll hear from Helen, her work incorporates multiple disciplines that go well beyond neuroanatomy and neurocircuitry. I think, what is truly impressive about Helen's CV is the number of honorary lectures that she has given. It reads literally as a who's who of the honorary lectures in neurology and psychiatry throughout the world. And I certainly heard her last talk at the AOA visiting professorship and lectureship, the David Siegel lectureship in 2007. And Helen advises me that there's been some progress since then.
Thank you. I want to get my slides. I can. I'm really honored to be here and to hear that description, and it's a pleasure to now be part of Mount Sinai, and I'm extremely enthusiastic that we can do some really innovative things here. And I want to give you an idea of where this journey has gone in the study of depression, but to actually preview a bit of how the logic is of what we're going to do at the center here. That was really, it makes it hard to give my talk after an introduction like that. Thank you. Disclosures wise, I'm going to talk about work that uses investigational devices. Don't do this in your basement. But there is a patent on the work with depression, and I consult to the companies mostly unsuccessfully, given what's going on. But we'll talk about that a bit later. I'm going to talk about the work of my team at Emory, and the main reason to show you this slide at the beginning is so I don't run out of time at the end, but to actually emphasize that this is the kind of multidisciplinary evolution that it's taken to really do this kind of work. So it is, you know, basically a neurosurgeon and a neurologist and a psychiatrist with really picking imaging, electrophysiology, modeling, psychotherapists. I mean, it takes integration in a way where sometimes we've been better at it or not, but this is actually part of the master plan that these people need to be together. And to that end, we're going to construct here the Center for Advanced Circuit Therapeutics, kind of a name I pulled out of the air, quite frankly, when I was trying to describe what I thought we should call what I wanted to do. But our whole goal is to really advance precision neurosurgical treatments for neuropsychiatric disorders. If you've heard Brian Capel, one of the functional neurosurgeons, you know, puts electrodes in circuits, and he doesn't really care if it's a neurological problem or a psychiatric problem because it's one brain. So, you know, to have a surgical partner that actually thinks that way is a real gift. And, but that beyond that, we're going to look at how we can leverage this explosion in technology and neuroengineering interventions and, you know, innovations, because we want to restore mood, motor, cognitive functioning. And that's broad. And so instead of trying to put it into silos of disease, let's not do that. Let's take what we do. Let's look to see what are mechanisms of what we do. So let's take advantage of having patients who've got stuff in their brain that we can study, but also how can we think about what are we not doing well enough so we can lead to personalized approaches. And there are all kinds of ways that a research program can build on that. So what's the construct to even think that you would do this? And this is, you know, the neurological blueprint, that if you think about a certain set of neurologic and psychiatric disorders, they're circuitopathies. You can look through the lens mostly of imaging, but through the history of neurolocalization, that there is an organizational structure for diseases and disorders and behaviors, and you can lay out various people's maps. And actually, I would argue that if a disorder of interest can be organized in this way, it is got the possibility to be developed for a neuromodulation approach. So what do we have to think we can modulate, you know, a disease circuit, if you kind of just buy that overly simplified argument? Well, you gotta tell a surgeon where to put it. And so you need to kind of know the circuit. Don't laugh. I mean, people think this is like a dream that you come up with, sticking a small wire into some place in the brain, that it's sort of random. And I'll show you just how very unrandom it is. But you need to know where to stimulate. You need to actually have some idea about what do you want to have happen, both clinically and kind of hopefully biologically, but at least clinically. And then who are you going to do that to? You're going to do it to everybody with the problem? It's going to be a first-line treatment? You know, and when you're doing as much as brain stimulation, invasive brain stimulation on the relative range of neurosurgical interventions is probably one of the least invasive. For all of us non-surgeons, you're opening up somebody's brain, you're sticking something in it, and stuff goes wrong. So who do you do that to? So even the development of it has to be done in you know, extremely intractable patients to start, but not necessarily. So a starting point really has to be, how do you deconstruct? What do you want to have happen? Who do you want to do it to? What do you want to have happen to them? You got to deconstruct a clinical syndrome. And all of our syndromes really are laundry lists of symptoms. At least in neurology, we humor ourselves that we know something about the organization of different kinds of behaviors. In neurology, we have a list, and it's like Chinese family menu. You know, if you have low mood and disinterest and 5 out of 9 of a list of symptoms, and now psychiatry is into lamenting how many kinds of depression there can be because of the factorial combination of all those symptoms. In neurology, we know that, you know, we don't mistake a Parkinson's patient because they don't have a tremor. We don't call it something else because they don't have a tremor, even though it classically has a triad of symptoms. So we want to assume with whatever syndrome, can we start to organize the symptoms? Can we imagine that, you know, a circuit mediates a particular set of symptoms? And can, if you're going to be doing neuromodulation, maybe you shouldn't expect that all symptoms in a syndrome respond the same. And we certainly know that in movement disorder. I mean, you can get rid of tremor and rigidity and people still freeze and fall and they still have cognitive and mood dysregulation. And we know that. In psychiatry, we have to kind of frame it similarly. We also need to realize when you're in a place in the brain, there's a lot of traffic and you got to balance getting something good with what's the cost. And you can be in real estate that can be very narrow and bad things can go wrong even at the cost of good, and how to temper that and study it. So I think, you know, this bigger meta transdiagnostic strategy, I mean, you know, the most simple way to really look at it is the fact that all of these disorders involve some combination of emotion, cognition, intention, and action. And that, you know, you go back to the basal ganglia anatomists and physiologists. I mean, this last 30 years of building up this logic, all through the lens really of movement disorder, is that these systems live, they communicate with each other, but they do have a functional anatomy. And so one can start with that to look at it. And in Parkinson's, you know, there's a syndrome. The good news in Parkinson's is there's a pathology, so you can build out a circuit based on where the pathological defect is. And it really came down to building a model, understanding why were people making lesions in the thalamus. I mean, they knew that in the early 20th century, you could damage the thalamus and tremor would stop, and you could get to the thalamus without even having imaging. You could do it by skull landmarks, but it wasn't until people like Nayland Long and his team really articulated what are the physiology of these basal ganglia systems, and why do lesions in the subthalamic nucleus or the globus pallidus or different nodes in that circuit, why does that alleviate Parkinsonian symptoms? And then it took inventive neurosurgeon to say, well, if I use high frequency stimulation to find the spot I'm going to lesion, maybe I should rig up a stimulator and I can give high frequency stimulation and control it instead of just damaging the brain. So we're always working from this logic of the circuit. Maybe there was a lesion, work backwards to figure out what was that about, move forward with technology innovation, and this is just the repeated path. In movement disorder, they're very good at identifying the subthalamic nucleus. You can do, as you're entering the brain, you can measure with microelectrode recordings, and the sound of the subthalamic nucleus is different than above it or below it. And so you can use physiology to localize what you started with, looking at anatomy. And you can look at these patterns of when you turn a stimulator on, you attenuate a 20 hertz beta oscillation and you turn it off, the tremor comes back and the beta oscillation comes back. So you can even have a physiology real-time readout of a symptom changing. So this is, you know, the gold standard. People have gotten really fancy. Instead of just recording off the subthalamic nucleus in the operating room where you're stimulating, you put in an extra recording device on the cortical surface, and suddenly you find that it's just not this 20 hertz beta, but it's linked to a high frequency gamma, and it's coupled. And what have surgeons moved on? They said, gee, when I can stop the beta oscillation and the tremor and the rigidity, but as a patient is receiving medication or receiving the DBS, sometimes they get dyskinesias. They start having too much movement. And Phil Starr actually realized that when a dyskinesia started to come on board, there was this little gamma spike. So actually this team has just published the first closed loop system where they've got the DBS going conventionally for Parkinson's disease. They've got recording going on in the cortical surface. They're measuring it off the battery pack that delivers the stimulation but has a prototype recorder. And as the brain is monitoring and it starts to see a gamma change, and that's when a patient has a dyskinesia, it turns the dose down and attenuates the gamma. So it's this example of where we're all going is you need to identify an abnormality, and maybe you deliver DBS all the time, but actually there can be a signal that you can attenuate and you can start to actually do very clever things. And we're just following these first early steps. So let's talk about depression, because I wanted to introduce you to the idea of the movement disorder, because what we'll be doing here is not only doing things in psychiatry. I mean, no one would invest in a center if all we did were, handful of these very interesting depressed people. But the idea is that just like the dyskinesia, movement disorder patients have mood instability. They develop OCD. They can develop cognitive problems. We don't do anything for them with the DBS electrode, but kind of looking at, these circuits, how can we leverage what we know about in psychiatry to those patients and vice versa? How do we set up so psychiatry can inform neurology together, not just, oh, my Parkinson's patient has depression, let me refer. That we do this in an integrative fashion, not just because that's better for the patient, but actually we can develop things that maybe we should be treating things simultaneously in the same patients. And how can we set the stage for that here, which I think is a unique opportunity. So this idea of depression being modeled like the Parkinson's story, actually this started, the inspiration, which I didn't realize, happened when I was a resident, because I did journal club and read a paper in 1983 in Brain about a compilation of a series of cases on stroke that were done in a collaboration between University of Maryland, Tom Price, and Bob Robinson at Hopkins, a psychiatrist. And they were looking at this phenomena, post-stroke depression, that develops in certain patients after stroke, not in everyone. And Bob was interested in ferreting that out. And in a series of consecutive patients at University of Maryland, Baltimore, in the ER, they would follow up with patients to see what kind of, who developed depression after stroke, and was it associated with a particular lesion. And Bob described that the most likely group to develop a post-stroke depression was in the left frontal lobe. What was lost in that paper was it wasn't just the left frontal lobe. 60% of people who developed post-stroke depression had a lesion there, but 17% had lesions in the parietal lobe, and 13% had lesions in the right parietal lobe. So it wasn't even just a left-sided phenomena or a left frontal lobe phenomena. In psychiatry, that left frontal thing was pretty big. And if you combine that, so when I left here and I went to Hopkins to do imaging, I met Bob Robinson. So I was like, meet Mick Jagger, meet Bob Robinson, kind of equivalent. But then I actually also met Malin DeLong. And if you think there was inspiration for thinking about these systems, it was him. I'll tell you the story. When we started thinking about Parkinson's depression or stroke depression, and I did that work with Sergio Starkstein, a neuropsychiatrist from Argentina that was working in Bob Robinson's lab. And that became the partnership. But that Malin wasn't thinking about non-motor loops at all. In fact, when I went to show him our Parkinson's depression work, he goes, "Oh, we kind of made that up. I mean, we study motor systems." But that logic of these segregated loops inspired other anatomists, physiologists to study the non-motor components. And over time, studying stroke, Parkinson's, Huntington's, I mean, I stayed very clear of primary depression until it was very clear that there seemed to be a signal that respected this logic. And then we kind of moved into regular depression. And we started to build our own little organizational structure based on what brain changes could we see in depressed patients. So we're doing PET scans, we're doing glucose metabolism, we're doing very simple-minded analysis. I mean, we were accused it was blobology. Even Bud Rowland, I think for a while, like what the point of these scanners. But over time, it became clear that if you started to look at how treatment affected these abnormalities, we started to see a more complex and evolving pattern. And, you had the people who were pharmacologists, you had the people who were therapists, they referred to each other, sort of. Everybody, wants 100% market share. And we said, we don't care. If you get your patients well, give them to us. We want to see how the brain changes. We started to see that meds work very differently from therapy. And we also started to get the idea that maybe people didn't get better on the same things, that there was something fundamentally different about these circuits that predicted where you went. And that's another topic. But in the story of thinking about, there's a lot of nodes in a circuit. So if I said, if you wanted to take the circuit in somebody who didn't get better, where are you going to put it? Like where's the hierarchy here? We don't really have a clue yet. But there are these patients that have this, don't get better on therapy, don't get better on multiple drugs, don't even get better on ECT. What do we do for them? And is there a clue in the nature of their symptoms that might help us to understand not only where to go, but what we want to change? And I think with everything that's gone on in the news the last week, with all these suicides and all the discussion, reach out, talk to somebody, like people should be asking themselves, what is the symptom that might make somebody hurt themselves, kill themselves? What is it that you want to escape that is so dark, so uncomfortable, and that you can't imagine anything else? That's the thing we want to figure out, and that's the thing we want to turn off. And I would like to make the argument that if you listen to a patient who has failed all treatments, what do they say? This, you can just read, and I've highlighted the neurological words that I find personally interesting. When you listen to William Styron, when you listen DBS patient 901, who actually is department chairman in an unnamed medical department in a nursing department in this country, who came to us. But it's to be in pain and not be able to move and cannot imagine anything beyond where you are right now. That's a very weird space, because if you have pain, you usually walk away. Someone says something you don't like, you walk away. We all know that exercise will attenuate a bad mood. This is an aberrancy of a system where you cannot move away from something that feels horrible. And when you do move, you lose yourself. I don't even know what that means. I just know that when I hear that, it's almost like they have an anosognosia of their own soul while they feel sick and can't move. I'm hearing mood, motor, and self, and there's a lot of data on that. So I've kind of reframed, because I think that fundamentally is why we should put things in people's brains. And if people don't have that kind of psychic pain, it's not about just being anhedonic, low positive mood. Something else might be useful for that. But to me, the motivation at the beginning was this psychic tension. We had studied it. You can make people sad And one of the areas that downregulates with successful antidepressant treatment actually activates when you provoke even healthy people into a negative state. And that's this region, hold on, let me get this working. Area 25, the subcallosal cingulate, that activates. It's a visceral motor efferent system. It is part of the computation of, is this good for me or bad for me? Its projections are down to the brainstem, the nucleus accumbens, the motor system. It's actually how you tally and move away. And because that activated with negative mood and depressed with recovery, and there were a lot of other regions, it's like, why not see it that turn negative off? And other things that are connected to it will recover. So you could plan a DBS surgery. You could kind of aim at that PET scan blob. We hedged our bet. We did PET scans before and actually had that this group of treatment-resistant patients had high activities, PET scan, blood flow scan read as higher than controls. And the frontal cortex is decreased. So it's almost like when your limbic system is up, you turn off your ability to move and think. And everybody knows what that feels like. It's just you snap yourself out of it. You go, I gotta get to a meeting. Okay, sorry to read that news. I feel bad, but next. These systems are constantly toggling on and off. And what do you do when they get stuck? And I think that was our simple-minded logic. If you can't talk it, drug it, shock it, put this electrode in, do high frequency like they do in Parkinson's, that blocks the activity. We should be able to shut that down, and the rest of the brain will kind of, they'll be disinhibited. What's ever holding it down will be released. So we did that, and we did it exactly like we did in Parkinson's to see what would happen. And here's a Hamilton Depression Rating Scale. These people, over 20 is considered severe. Here's where they start. Here's what happens with some stimulation over a week in the hospital. Here's what happens when you turn the stimulation off for a week, turn it back on, and over 6 months that these people who were ECT failures, on average 4 years in the episode, having failed every treatment anyone could think of, 4 out of the 6 got better. This is what's encouraging, at least the logic is correct. We expanded to 20 patients and actually saw that, yeah, they got better, they stayed better. So similar kind of group. I moved to Emory. We looked at not only unipolar depressed people, but people who have what's called bipolar type 2 depression, where they have not pure manias, but have mostly depressions, but are very sensitive often to medications and can become hypomanic. So they're hard, it can be hard to treat. They all got better, but we didn't do quite as good as we had done in Toronto, but it was a new team. But this was in an experiment, the opportunity to try to figure out, and you finally get some non-responders to kind of get at what are you doing right, what are you doing wrong. You know, it's always scary if people are doing too well. But I was reading something the other day and someone says, oh, a depression score went to 0 really fast in everybody. I don't know, I'm a nihilist, I don't believe it. You want to believe it, because in order to even get to this stage to move on, you got to get lucky because you got outliers that responded. But you don't get everybody well. So the question is how to get a handle on what that is. And I'm being prescient to that a little bit because it was very clear we didn't know. We were doing something right, but we didn't know what we were doing. And no one was listening to me saying, we don't know what we're doing. And this moved to an industry-sponsored trial way too fast because we had good results. The surgeon knew where to put it. He was doing it really well. It didn't really seem to matter in terms of surgical targeting, who responders or non-responders were. And this is the BROADEN study that St. Jude Medical Incorporated did. And they planned for 200 people, 15 sites. They had a deal with the FDA to do a futility analysis at the halfway mark. It was 6 months of active versus sham. So people were implanted but not turned on. No psychotherapy, no change in meds. And basically, this is the blinded period. Active did not beat sham at the 6-month endpoint. And I just showed you in these open label and even our 1-month blinded studies, we were seeing response to 3 months. We told them, gee, go at least 6 months just to be sure. Total failure. But as you go, and then they turned everyone on, even though they didn't know what they'd been on in the first 6 months. And they got up to 30% responders in everybody at a year. But they continued for open label, long-term follow-up, making adjustments. And at 18 months of active stim, they actually had a 50% response rate. And at 24 months, that was pretty much sustained. And these people have sustained their patients out that started in 2008. There's something here that everybody missed. And the only thing that explains in the analysis of the data, the recovery over time, and these people are on average 12, 15 years in episode. These are much more chronically ill patients than any of the open label published studies, was changes in the location of what was being stimulated along the electrode. So I just wanted to start by saying, you know, this is hard, even interesting, logic-oriented things in the hands of committed researchers in a place. It tends to go well. Everybody, you know, this experiment went from don't believe it, yeah, it's obvious, gee, it's not your idea, to, oh yeah, when this failed, we knew that was stupid all along. Okay, so where are we? This is where, you know, really the rubber meets the road experimentally, because if it was easy, we'd be done already. And the thing is that we've got now patients 11 years at Emory. It became hard to leave Emory because I've got a cohort of patients, 35 patients, not huge, that continue to do well with an electrode in their head. These are people that were disabled, ECT failures, stopped working, have gone back to work, have, you know, not everyone is, you know, Miss America, but that people are living without wanting to be dead all the time. And they are functioning in a very different manner than they were with conventional treatment, even ones that had been doing well. So we have kind of an obligation to figure this out. And if you study depression, my attitude is this was my best chance to understand what the disorder was by understanding kind of what we did. But if you're trying to get something to be available widely, then the question is we need to figure out how to help people to do it right. So it's not just one surgeon or a few surgeons that know how to do it. It has to generalize. And I know for the surgeons in the room, that's how it is. I mean, you may be the developer of something, but if you really want it to have impact on patients, you can't be the only one who does it. So we're back to what I said at the beginning. Who do you blame was how this discussion went about, but it's really the basic principles of, did you miss where you put it? So is there some variance in placement that's more important than you realized? Did you pick the wrong patient? And that's always what we say. That's a really hard problem in psychiatry, any more than it's a hard problem in internal medicine. How do you decide who gets what drug? You don't know either. I mean, only infectious disease knows, and cancer now knows, because they've got some biomarkers. But the rest of us is clinical judgment and trial and error. And then we can blame industry for their design. No, we have to help them to do the design better. So what's and then the age-old argument is it was all placebo. And I want to convince you that placebo may be part of it in the upfront, but that we can design experiments differently to show that a sustained response over many, many years is not placebo, or a magical transformation in people who have been so chronically ill. Or, you know, I guess you could say it's like trephination. I mean, just put a hole in their head and these people got better. But the reason it doesn't seem to be is, well, first of all, the Toronto team, our team, their teams in Spain with small samples, if you get better, you stay better with ongoing stim in this target. So that's encouraging. But if you do a blinded discontinuation in people who are well, you actually see a loss of effect. And the good news is that in this particular target, it's over weeks. Actually, I just talked to my team and they said that one of our patients who's been in remission, she's a pharmacist, she had to go to the cardiologist and had to have the device turned off to get her EKG, and she forgot to turn it back on. And so she realized that it was off like 3 weeks later because she woke up and she sort of had the dwindle. She had trouble kind of getting out of bed. So this is not a, like you aren't, this isn't like a stimulant crash. And other targets in the brain actually have that phenomenon. This is a slow slide. And the best, I think, evidence that this is not placebo is like the patient I just said. These batteries, which are going 24/7 right now, they have a shelf life. They need to be replaced. There's now rechargeable batteries, but that these are data on that Hamilton depression score. So low is you're not ill. And then this is the point at where the patients came in for a battery replacement because the device had died or they had a complication and had to be, fixed. And you can see that as you replace the battery that you regain the antidepressant effect. So I think placebo will have to be mitigated in terms of a clinical trial design, but this is not, just we need fake surgery and the theater of surgery will be good enough because that doesn't seem to be the case. But I think, back to the neurology, the real point of who gets better and who doesn't, given that you can find the right patient, but let's just leave that out for now, is the where. So the first obvious thing is figure out where the variance is in terms of where's the place you're stimulating. Maybe you just missed the bullseye. Well, it turned out that the anatomical location of the active contact and people got better or people who didn't, both in Toronto and in Atlanta, could not explain the responder non-responder difference. So everyone naturally said it's the psychiatrist's fault. They need to do better on who the right patients are. But in fact, if you looked at the PET scan changes we had in our very first paper, here's the PET scan changes, the blood flow changes going from pre-op to 6 months. Responders and non-responders both did what we wanted, decreased activity at area 25. But what you can see, and particularly if you're not a neurologist, to follow the yellow circles and just compare the responder to non-responder, things that were away from where the electrode was, remote changes, things that 25 was connected to, did not change in people who didn't get better. Well, you can make an inference about that. Well, if a remote area, 25 is talking to other areas, how does it talk? It has connections, it has axons. We can know the track tracing characteristics by studying that in postmortem and in monkey brains. And we knew even when we started that this spot was connected to various other areas. It was kind of at a nexus, but we didn't have what we had later, which we didn't have diffusion scans. We couldn't do tractography to map it. But in Atlanta, we did that from the very first patient. And what we then did, Ki-Sung Choi, who is here with me, is in radiology, and he'll be in the center here. He's a signal processing engineer and an expert in tractography and white matter mapping. We basically said, and Patricio Rivapose, who was our head psychiatrist, and Cameron McIntyre is an engineer at Case Western who does cable modeling to understand DBS kind of in theoretical models. We basically all got together and said, let's take our tractography maps and let's see what we do alike using Cameron's models, Ki-Sung's images in these patients based on how they did, and said, there must be something that we do in common between everybody that got better at 6 months. So let's do a common map and see what the connections are. Not on average, what do we have to hit in everybody? And here's the map. And then we said, okay, what did we do differently in people who didn't get better, knowing that actually a non-responder might be non-responder for various reasons? Well, that's the red, and the orange is the overlap. We got most of it. And the non-responders weren't total losses. I mean, they got partly better. They just weren't where we wanted them to be. And it turned out that we hadn't reached the full compendium of white matter connections in the non-responders. And it's like, okay, well, how do you prove that? Well, we had people that got better between 6 months and 2 years. Then we got into the kind of arguments. The psychiatrist said, well, I tweaked their medicine a little bit. I did it. It's like, could be, but we also did trial and error and changed the location. Let's just ask the data. If you went from a non-responder to a responder over that time, where did the track end up? Again, everybody has to share. And it turned out we got into the right place, completed hitting all of these bundles, and we could see why we did the improvement. People still weren't happy. So what we did is we said, here's our template. You need to put that electrode in the place that hits the cingulate bundle, that hits the forceps major, well, it's minor actually, the connections down to the mesial temporal lobe along the uncinate fasciculus, and also these small tracts that are very hard to see that go down to the accumbens and the thalamus. We can literally drive around on a single subject's tractography map, hang out in the OR, sit there with a laptop, drive around until we get to the spot that hits those 4 bundles. And we tell the surgeon, we tell Bob Gross, right there, I want the second contact on the lead so that we've got play to stimulate within that space. So we could do a tractography guided targeting. And then what we did for 6 months is we never changed the location. And we had a 70% response rate. So without ever changing anything about the patient we enrolled, using the imaging, using that logic, and then testing it, we could accommodate most of our variants. And so now we can kind of work on how to proceduralize that better for others. Now, the other thing we can do is say, well, maybe those people still don't get better is because they really needed a different kind of intervention. And I alluded to that. Not everybody with very bad depression has got that psychic pain and can't move. Some people just have almost a more mood lability or have a primary anhedonia. And it's something we can test. And Martin Fagia, a psychiatrist from Amsterdam, was recruited here. He'll be here also. He's here. He's actually worked with stimulating the nucleus accumbens. So we have a team like no other, where instead of, everybody's experienced with what the target is that they use, and, there's so much development time, we'll actually have a team where we can really develop a program where we can work in different locations because the team is comfortable with that. So I'm kind of excited about that. The last thing is the notion of what did we learn what else can we learn by watching the trajectory as we got better at the targeting, which is a totally kind of anatomy-based, circuit-based logic. Well, if you actually look at how these depression scores changed over time, the red line was the first Emory group when we just did it by anatomical targeting. And the recovery curve is sort of linear. And that's kind of what everybody figured. It kind of takes a while to get better from depression. But then when we started to do the tractography targeting, that's the green line. Actually, what I didn't tell you is in the operating room, we actually turn it on. And it turns out we had noticed from the very first patient, people read all about this, it's the coolest part. You're kind of stimulating the operating room because you want to make sure nothing bad happens. And you'd kind of get into one of the contacts and you'd stimulate and start turning the current up to see if something bad happens. And patient would go, oh, I just feel lighter. The void is gone. I'm not drowning. I'm out of the hole. They would say all kinds of very mystical, metaphorical, pain was gone. And sometimes you could reproduce it, sometimes not. You would see it in one contact and not another. But over time, we realized that that was tracking with when you were in the right spot based on the tractography. So it now is a biomarker clinically of when you're in. And it looks as though if you give stimulation in the operating room, the magnitude of where your depression rating scale is out of the operating room for a month with the stim off is actually maintaining. This is acting to me like we're cardioverting an arrhythmia in the operating room. And then we're putting in a pacemaker to hold it. And I'll tell you now, we are going to prove that. Nobody likes this idea so much, but it certainly fits with what we're seeing. And there's certainly logic in other models. I mean, these people have got chronic heart disease, maybe an MI. They've got a chronic state of their brain that for some unknown reason, they were previously responding to ECT, and then they have a malignant transformation, and now they're non-responsive. They've got an arrhythmia. We're going to flip them, and then we're going to hold them. And I think we can set up a logic stream to test that. And we have evidence that this is happening on our PET scans. In the old days, we didn't know that we needed to take measurements early. 3 months, 6 months, decreased area 25, corrected frontal cortex. Game over when we looked. What we did at Emory is we said, let's look at a month off after surgery, after a placebo month, and then let's look at 1 month and 6 months. And what we found is the local affected area 25 and the connection along the uncinate fasciculus to the medial frontal cortex, game over. That's an effect from the OR. It carries over a month after surgery. Here you can see that behavior with a region of interest analysis. It goes down and it stays down with ongoing stim. Whereas changes that are happening in prefrontal cortex, remote from what's being actively stimulated, no change by being exposed initially, only changes with chronic stim, and actually you lose the effect if you're not in the right spot. So you don't get this stimulation-induced plasticity. Is that because of the electrode? Is that because of the electrode in rehab? I think that we've got an opportunity to figure that out. But we also need to model this notion of we have to think about what we did in a totally different way. When I've shown this picture, I have physicists go, oh, you have a bistable system, and that's a cardiology approach as well. I think that we have computational modelers to kind of look at this, and we have other ways to study it. And the same is true in things, in other disorders that do DBS. Parkinson's, you stop tremor and rigidity fast, but you actually have changes in gait that take a while. OCD the same, depression the same. We've got to model this by having patients come back, being scrupulous in that operating room, because you may miss what you do if you don't study it in the OR. So A, I'm sorry, it'll take more time in the OR, but maybe we should be doing our main treatment in the OR. I mean, I used to get into fights with the surgeons. No, you can test that outside the OR. Then we couldn't find it again. We didn't even know it was important. And so, you know, we've got to be observant and iterative. So I know the time is short. We got started a little late, but I want you to also appreciate that we've got to come up with new ways in which we track. And that now we've got this device implanted in people like I showed you at the beginning where they were studying dyskinesias. We have this device. We're tracking the depression signal over time, all the time, to see how, is there a signal on area 25 electrophysiologically? Is there an oscillation that changes? We'll have high density EEG in the lab. So when patients come in every week, we can be tracking where they are. We've got teams of people lined up, so we'll have a performance lab, that things we do in the lab, things we do in the field, partnering with people all over campus and not on campus to track these things. And we've already got clues. I just want to show you one thing that is interesting back to the neurology, that where I think the beauty of being a neurologist in this is that you really do see what we're seeing differently. So what do we know? You know, Darwin studied facial expression. I mean, we need a metric. I mean, what motivates this? We need a metric. Here's this Hamilton over time. Hamilton score is down. What is going on here at 3-4 months? You get this instability of the Hamilton. They losing the effect? Should I turn it up? Should I get them more therapy? I mean, what is it? And the clinicians know it's generally a rough patch. You know, their families start to saying, you've had 3 months, get a job, do something. You know, what is integration really back to the real world? It is hard if you've been sick a while. And are we seeing it reflected in an imperfect measurement? In a Hamilton depression score was designed to study depression in the hospital, not 6 months, not a year, not 5 years. Look at the pattern of different patients. Some people get these instabilities. They get it in different ways. We need to know, stay the course, get more therapy, work harder yourself. No, we need to do something. Maybe the device will tell us, but maybe we need a better metric. So what do we know? Here's one of our patients. She was happy to have me show this picture. Here is actually Darwin with Duchenne's, you know, poking people with electricity on their face in different muscles to create different emotions. This is the suffering emotion. That's this lady, and she described her depression as a virtual paralysis. Here she is a year later. I mean, it's like she looks like, you know, 20 years younger besides just the capacity. And we got interested in, you can see this change in facial expression happen on the operating room table. I could see it. I didn't know what I was looking at, but there's a way people's eyes start to change where I know they're going to say something. I don't know what I'm looking at. And then you think about, what is the neuroanatomy of facial expression? Neuroanatomy of facial expression is complicated. Middle cerebral artery stroke, contralateral face is paralyzed, but mimetic smile, someone tells a joke, it's symmetric. Get a cingulate stroke, you're slow as hell. You move like you're paralyzed, but you move symmetrically. And if someone tells you a joke, you have a very distorted change. Where's the anatomy of emotional or spontaneous facial expression? It's in the midline. And where do we have our most profound finding that distinguishes our DBS patients from other kinds of treatment resistance? A disconnection right there. So maybe facial expression is the window to the kind of depression and a readout. So we got a computer scientist to take the videos that Andrea Crowell, one of our psychiatrists, was videoing because patients said cool stuff, so we wanted to have it. Well, actually, let's use the videos. So Har went through and did what a lot of people do with videos. She cut out the face, she put it into some math thing, looked at different kinds of measurements of each voxel along these different videos, and came up with 42 features of movement in these videos over time. And actually, we've got the capacity to say, let's look at what the face is doing in Sahar's algorithm early when your Hamilton is high, and we trust that sick is sick, versus late when you're well and the Hamilton is low. Can we model and get a signal of the facial expression with this kind of machine learning algorithm? We can. And then said, let's test it at that point where nobody knows how to make a decision. It's 3 months in and your Hamilton spikes. An algorithm in a clinical trial will say, make an adjustment. We actually think that's a bad idea. How do we prove it? We ask the face, are you sick or well? More than half the time, the face says you're well. Stay the course. So in fact, we may have a biomarker using machine learning on this, and we'll test it out. And we've got partners where we might do this in movement disorder. What is hypomania in Parkinson's? Is it the same kind of abnormality or something different? What about a depressed Parkinson's? Are they showing this depression kind of signal or something else? We can do motion capture and study axial movement. We've got steerable leads so that we can go into these different systems. So if we set up the model of our new center to where people have surgery, we have integrative care, we have the research lab right there. We can do experimental trials or do clinical care, have a lab where we test people, we can look at the imaging, we can look at the electrophysiology, and we can do whatever we want because we're iteratively doing it together across platforms. And that's the plan. So here's the beta team that'll all live here on the 10th floor for a while, if we get there. And hopefully this was okay for you. But that you will all meet Brian Capel, who is the primary functional neurosurgeon, but there's 2 neurosurgeons here now that are working in epilepsy, and we'll all be joined. And everybody will actually live here together and come up with a fun and inventive and hopefully successful way that we take care of these patients in the maximal way, do interesting new things, and take care of the patients. And I just want to end by thinking about the patients, because no matter what we do in this space, we have to work out all of these other pieces to do a good job scientifically. But for depressed patients, you never can forget this part of it. And we have to train patients to know if it's in movement disorder, if it's in psychiatry, what part does the device do? And what part does the patient do with a trainer, with a therapist, with a rehab expert? And we got to know which one we do and which one they do, or we will fail no matter how good our science is. But I think our new team will be in a good position to do this as well as anybody else can. So thanks.